Hi, everyone. Thank you for being here. This is really Stefano's presentation. I'm so sorry that I have to be that you're stuck with me like for this work. So AlphaHOGs. So I am emphasis on the towards. Okay, so we've seen like this revolution. We had like really a fantastic talk this morning by Martin Steinegger, who really gave like the overview. And as you know, almost everything we know at the molecular level is projected from experimental data that are just concentrated in just a few species. So how do we really take just this little information and characterize life? I mean, it's actually a daunting challenge. And for that, of course, evolution is key. I don't have to tell you this. And very early, already 100 years ago, people were realizing actually at the molecular level, there's just so much conservation. And of course, you know these quotes, nothing makes sense in light of evolution. So we have a chance maybe to do that. And actually, this, I also want to mention a quote from Yanis, who's in the audience. So the hogs, a really important concept in our lab is the concept of hogs. Is a projection of gene trees. So we have gene trees here and species trees, and you can see. Well, you could infer the gene tree, root it, infer the duplication and speciation node, and then you will have a pretty good idea about the repertoires of genes across the different ancestors. We like to work with some projections in terms of this nested orthologous group structure called hierarchical orthologous groups. And so, for instance, that are kind of bag of genes that are descending from one ancestral gene at each level of interest. And if we can do that at the scale of all genomes, then we can reconstruct our ancestral kind of gene repertoires. So here, for instance, we have a duplication of a gene here in some vertebrates, and so we have, well, actually here in some mammals, and so we have two copies. So we have two hogs at the level of the mammals, but it's only really one hog at the level of here of the tetrapods. So now the question, and actually, I mean, I'm sure many of you are thinking about this, is how can we now use structure to go a little bit deeper in the tree, maybe deal with like the issues of the twilight zone. We heard that also this morning in terms of the significance, but also maybe for other part of the orthology prediction. And so in our lab, we've developed recently a method called FastOMA, and so I don't have time to get into detail. Actually, Sina is really like the main drive behind this project. But like many orthology methods, we first need to do some homology clustering, essentially finding the different bags of genes that are homologous, and then we infer some trees and in the second step. And well, maybe we had a very nice talk by Anshu earlier, showing that, well, sometimes orthology prediction is quite challenging. With FastOMA, we're really happy about the speed up that we managed to get. And I mean, there's a preprint on this, but I mean, if you compare with the traditional OMA algorithm, if some of you have been in the past frustrated about the speed, which we also have, and there's quite a big difference now with FastOMA. But now, in this context, we can ask ourselves, okay, how can we use now structure? And so, indeed, for the cluster homology, we could use potentially structure, and then for the tree inference. So I will not talk about the tree inference using structure this morning, I mean, given the length also of the talk, but there also I want to plug another piece of work, Fold Tree, which actually works also with the 3DI alphabet that was discussed earlier, and actually which shows that we can build actually a pretty good a structure tree. So if you're interested in that, you can have a look. But I want to focus, I mean, actually really Stefano has been focusing on the homology clustering. And so the way we do this, and again, it is one more building block, the last building block I would like to introduce here, which is kind of a fast placement into families using k-mers instead of, and this has been also a topic within EvolCompGen. We've had a number of really interesting methods. This one is trying to index the k-mers in terms of the hogs, in terms of the families, which is also has some advantages in terms of scalability. And so, for instance, here, if I take this globin family, which here in the case of the vertebrates comes in more than 2 family, but you have, myoglobin, which binds to oxygen in the muscle, and hemoglobin in the blood. If these are like, just for example, with this k-mer, this actually is not even real k-mer, but just as very simple here. The way this is then indexed is we have kind of like the k-mers that are at the level of the entire family, but then the ones that are specific to some subfamilies. And so in principle, we should be also able to use, for instance, the 3DI alphabet, and then use kind of structural alphabet for these fast placements. Okay, so does it work? So again, this is still very preliminary, but essentially we've picked like 4 species where we mask what we know about them, and we try to place like the sequences. So there's one easy one, which is human, because we have a lot of closely related genomes, and there are 3 that are much more challenging because they are they don't have any close related species. And what you can see is just in terms of like the placement into family, the overlap is a bit moderate. So the question is this a good thing or is it a bad thing? And well, if it were completely overlapping, then that would certainly be a bad thing, because we could not glean any additional information from the structure. And here I should say, as a reference, we compare that to alignment-based kind of clustering, which we think is still kind of, I think for our intended purpose at this stage, kind of gold standard. Obviously, if you want to go deeper than alignment-based, we would need a different benchmark. Again, it's just a first step. And so here you have kind of a precision recall curve, and you can see actually 3DI, so the structural alphabet is doing a little bit better if you're satisfied with relatively moderate recall. If you really want to push the envelope in terms of recall at the expense of precision, then it's a bit worse. That's what we observe also in the other data sets. And so now the question is, can we combine? Or can we? And so there, Stefano just tried something really simple, essentially kind of summing the scores that you get with the 2 schemes. And interestingly and encouragingly, it looks like now the curve that we can get, is really the best of the 2 worlds, at least for this setup. So, well, to leave some time for lunch and perhaps 1 or 2 questions if we have time, I'll stop here. So, first few steps, it's encouraging. I'm sure that this is on the mind of many of you. Would really love to talk if you have also some ideas. Well, so far it looks like combining amino acid and structural alphabet k-mers can, recapitulate better what we do based on expensive kind of alignment-based techniques. And we will need really to face the question about how do we benchmark kind of like the deeper prediction, the things that don't even agree with alignment-based comparison. So I'd like to thank, well, in particular, Stefano, who's here, the rest of the lab. And there's 2 posters that I really encourage you to visit alongside, of course, all of the other poster of EvolCompGen over lunch. And thank you all for your attention. And thank you to the organizers also. Yeah.
Any questions?
So you're looking at 2 different alphabets for k-mer placement, and I don't have a good insight into exactly what the long-term outcome of that is in terms of, but your maximum on your recall line was 60%. So it seems like even your best method's doing really badly. And should I worry about that?
So, of course, you should worry, Danny. These are like for the I here purposely focused on these three kind of outgroup species for which we do not have much, close. I think it's tricky, even for sequence-based comparison. We even know that we have homologue missing between human and mouse, believe it or not. I mean, this has been reported. So we're really not doing a good job overall. At least we're doing, in relative term, a little bit better. I didn't show the result for human. In fact, it just occurs to me that I cut slides a bit too aggressively, but the human results are extremely good, and this is because we have a lot of, other mammals and primates. So, it depends on the species that you're looking at. I wouldn't focus so much on the absolute level. We know, we're hitting the wall at some point when we are really going back in time. And I think there's a great deal of uncertainty, both with the structure-based k-mers, the sequence-based k-mers, but also really the sequence alignment, frankly.
Thank you.
Thank you, Danny.
Thank you again.
Thank you.